patterns in cell populations to probe the causes and cures for disease. They lead the jump cell painting sense insertion. The title of her talk is Cell Painting Database on this topic. Anne Carpenter. Dr. Carpenter.
Pleasure to be speaking. My goal is, I promise today, my goal is to be very educational. I'm going to tell you not with respect to images. So today everything I'll talk about is public. The PDF of slides, I'll show them in case you would like to grab a copy, so you don't have to jot down citations. All right, so let's get started. So actually, I should say I've been understand a little bit of my. So actually, I should say I thought, well, where should I tell you that I? So States. I was a little girl, I loved reading very much. So of course, the career path I thought I should take is I want to be a librarian. But then I discovered that if you work in a library, it doesn't mean that you get to read all day. So I thought, okay, I need a more practical career path. I'll be a soccer player instead. Football player. That maybe wasn't also very practical because I wasn't very good at it. But by the time I got to junior high school, I didn't care about anything except finding a boyfriend. When I got to but by the time I got to junior high school, I didn't care about anything except finding a boyfriend. When I got to college, I at them. So I thought, okay, science, that's a good choice. It's has good career opportunities. So this will be a good option for me. And I definitely want to go to industry. I definitely don't want to stay in academia. I love school, but I want to have an impact on the world. So I thought I should leave academia in order to have an impact on the world. So you can see that's kind of funny. And when I decided I would need to go to graduate school in order to be able to lead science as a problems. I see that as my role to convert really difficult biology problems into something that is understandable by computer scientists and can really make an impact. And so as you can see, I thought I had to go to industry in order to make an impact, but I found that being in academia, I can also have the sort of impact on drug discovery as you will see in the talk today. Our lab focuses on images as a source of information, and there's several reasons why we like images, and I'll go through these next. Images of cells specifically. Oops, hang on. Oh, I'm sorry, I got a little bit out of order here. So let me give a little bit of background first, because I know some of you, biology is a bit of an unfamiliar area, but you might know some of these words. You might know that we all have a genome with DNA that encodes the properties that the human will have, including some diseases. Most diseases have some kind of genetic background. The genome is transcribed into mRNA. So a lot of biology experiments involve measuring mRNA, how much mRNA for each different gene is present in a given person or a given cell. And then the mRNA gets translated into protein, and the protein is usually what does functions in the cell. It's usually what carries out different functions. And it's usually, if there's a mutation in a person's genome, it gets transcribed and then translated so that the protein doesn't work quite right. It might have different function, it might have no function, and that's what typically is causing diseases. Now, when we talk about computational biology, it's very common for computational biologists to work on one of these three levels. They're all very linear, they're all very quantifiable. These are known as the omics, the genome, the transcriptome, the proteome, those are all omics. And they're kind of readily measurable and quantitative. They just match a computationalist's thinking very well. But what I want to emphasize today are cell phenotypes, the morphology of cells that comes a bit downstream of this. So you have all these proteins doing things in cells, and they work together to make the cell have some function. It makes your liver do what your liver does. It makes your heart pump. It makes your brain think, and so on. Those phenotypes, those morphologies, I think are really valuable to measure. They're not as easy to measure. They're not as discrete. They're not as linear. But I think it's well worthwhile to focus on this level, because this is the level where we can really see the impact for patients that have different disorders. So that's what we'll focus on today. And if I can summarize almost any biology experiment, no matter how small scale it is, biologists typically will be studying a particular gene or a particular small molecule. That's a drug or a compound is what you might call these. So they'll pick something to study. They'll perturb it in some way. They'll add the gene to the cell. They'll take the gene away. They'll add the drug to the cell. They'll add combinations of drugs and so on. So you take some kind of gene or small molecule, you do some kind of perturbation, and you have to choose a time point or a cell type or a cell model system that you want to study it in, and then you observe and measure something. That's basic biology experiments usually can be mapped to this kind of thinking. So some kind of input, some kind of output. That's really what it comes down to. And you might ask, what do we measure? And the answer is measure all the things, because there's so much that can be measured. And we already talked about some of the things that can be measured. You can perturb the cells and then measure, did it change transcription? Are there different mRNAs present? Did it change translation? Are there different amounts of protein present? But as we can see, we can also measure cell phenotypes, and that's what we'll focus on today. I want to talk about how microscopy can power drug discovery. So first, I want to give some kind of short overview of what is the field of drug discovery, how does it typically work? It's maybe not as complicated and not as successful as you might think. And then it's a field that really needs disrupting. And so we'll talk about microscopy as a way to potentially really accelerate the drug discovery process. All right, so let's begin the overview of drug discovery. Sequencing, genome sequencing, has been uncovering the causes of disease. And this map, I think, is just amazing to me. It really is a triumph of humanity working together to sequence the human genome. Each of these linear items is a chromosome, a human chromosome. And in the early days, in the 2008s, we knew a few genes that caused a few disorders. But over time, this number has just absolutely exploded. And this map only goes to 2017. You can only imagine how more full it would be now. Usually for many disorders, complex disorders, we know hundreds of genes that underlie the diseases. So we're learning so much at the genome level. And we've made all kinds of different technology breakthroughs that overall should make drug discovery better and faster and cheaper over time. So DNA sequencing, we already talked about, orders of magnitude improvements over the past few decades. we can also chemistry. We can also synthesize drug-like molecules, again, orders of magnitude more quickly than before. We can calculate protein structures, and even in the past couple of years, we can now predict these computationally with AlphaFold, and so many other advancements, so many different things we can do. And humanity as a whole spends a lot more money on research and development for pharma over time. So all of this together should make drug discovery better, faster, and cheaper. And just like in the computer industry, you, I'm sure, are familiar with Moore's law, where compute gets cheaper and cheaper over time. Keeping in mind this is a log scale, so just dramatic improvements in the cost of compute. What you might not be as familiar with is Arum's law. So it's literally Moore spelled backwards, because we have the exact opposite relationship in drug discovery. Despite all of those beautiful technology advancements I just described, drug discovery keeps getting harder and harder and more expensive. So if we look at the number of drugs, new molecular entities, the number of drugs that are discovered per $1 billion, it's decreased from, in the 1940s, if you had $1 billion to spend, you could get 30 new drugs on the market. Whereas today, it barely, in the same price adjusted, time adjusted dollars, today the same amount of money gets you only one new drug out. It's $1 billion to find a drug and to get it clinically approved and marketed. So this is a really depressing industry to work in, I have to say, from this big picture point of view. And it's not just the money, it's the time that it takes to get a new drug onto the market. So pre-discovery is where most of us academics are working. We find somewhat promising things, we do screens, we kind of narrow things down to the point where eventually right here is where a particular drug is chosen to test in humans. And then it can be many years before it eventually gets to the market and then actually helps people. And so the total time span tends to be on the order of a decade. So it's time consuming, and we're really bad at it as an industry. Drug discovery fails most of the time. So this shows across different disease areas, but on average, we get about 10% of the drugs that enter clinical trials actually are successful and, you know, treat the disorder that we wanted to treat, and they don't have too much toxicity. That's 10% of drugs that enter clinical trials, not to mention the steps before that where we think all these drugs are promising, they probably have a 1% chance or even less to make it all the way to the end. So this is, and the drug discovery rate is getting worse. Okay, so now you're wondering, why did I come to this talk? This is really depressing. Drug discovery is hard and difficult and expensive and time consuming, and it's getting worse all the time, even though we have all these technologies. Well, this is depressing. So we really need to accelerate drug discovery. This industry needs something disruptive, and I think that with AI and computer science, there are some ways that this is going to be happening, and I think the next few decades are really going to see a shift in our ability to discover new drugs. So I thought it would be helpful to explain, first of all, how do we wish we could discover drugs? What do we mean when we say we discovered a drug? Well, we take a chemical, we give it to a human with a disease, and we cure the disease, or we treat it at least. The human becomes healthier than they were before. So we wish, if we really want to know if a compound is going to be effective in a disease, this is the experiment you have to do. This is the only way to do it. And of course, this happens in clinical trials, but it's not very helpful to discovery. We can't just take 1,000 different chemicals, give them to 1,000 different humans and hope for the best, right? We know that the percentage success will be extremely, extremely small. So we have to make some kind of proxy, and this is where most of the field that I work on is working. We use cells as proxies for entire humans. So the nice thing about this is we don't have to just test like one at a time. We can test millions of chemicals, and many pharma companies have 3 to 5 million compounds in their freezers that they routinely are taking out and testing. So you can test all of them if you're using cells. You just take cells and you have to sort of make disease cells in a dish that you can grow and that you can add each compound to, and you can see whether it becomes healthy or not. So this is called an assay. Any biological system where you have where you're representing the disease is called an assay. And here's one example. If we take healthy human cells and we grow them in a dish and we add tuberculosis bacteria, they become infected. So there's the blue human cells and the green bacteria. And we add drugs one at a time to these samples of cells. And we ask, are there any compounds that keep the human health cells healthy, but kill the tuberculosis or cause it to not proliferate? And that readout, that phenotype, is the outcome. So the input is whatever chemical we add. The output is whatever readout we've chosen. And as you'll see, we've picked morphology as a readout we think is very powerful. Okay, so that's a little bit of a view on drug discovery. It's a little bit of an explanation for why we really want to improve the speed of the and cost of this field. Why do we think microscopy is a good way to do this, and especially the intersection of computer science and microscopy? So microscopy offers, first of all, now we're getting to those reasons why microscopy is so valuable. It offers single cell resolution. And many of you may have heard of single cell RNA sequencing and single cell proteomics and so on. These are pretty expensive techniques that allow us to measure mRNA and protein at a single cell level. But microscopy has been single cell resolution for hundreds and hundreds of years, and it offers an ability to look at the heterogeneity of a cell population, because cell populations are quite routinely heterogeneous. The second reason we love microscopy as a readout is we can examine things beyond just simple cells. We can look at all the different biology systems shown here, including whole organisms that fit into a teeny little test tube or a teeny little well, things like these little worms called C. elegans or zebrafish. They're much more similar to humans than organisms like yeast, and yet they still can fit into an experimental system to test at least hundreds to thousands of compounds, even if not millions. And so many of these model systems are a bit of a trade-off. You can try to get something that's more whole organism-like these guys, but you take it's much slower to work with these organisms, and they're bigger, so they take more materials and so on. Working with an organism like yeast, which is also eukaryotic, can be really efficient because you can do all kinds of clever experiments where you pool all these together, and they're very inexpensive experiments that can get you pretty far. Third, image analysis is quantitative. And I realize on this slide, there's going to be lots of words you don't know, but let's focus on the big picture concept here. Here, we're looking at a type of cancer and called a type of leukemia. And what we're trying to do is find a drug that causes the cells to look like this instead of their natural state like this. So these are leukemia cells that are growing out of control. They're very small because they keep dividing. Whereas these are leukemia cells that have paused in their cell division. They've gotten big and kind of grown to a steady state where they don't want to grow anymore. So our goal is to test thousands of compounds. We're academia. We can't afford to screen millions of compounds, but we can test thousands of compounds and find some that have this behavior. And so you might say, from an image analysis perspective, you can see the software has outlined each one of these individual nuclei of the cells. It's pretty simple software problem to outline each one. And if I asked you to draw me a histogram of what would the quantitative data look like from these images, you might draw something like this. This sample over here, the natural state of leukemia cells is kind of smallish. And then if you get to a larger size, a larger amount of DNA, these guys over here looks like kind of a big bump. In reality, the data looks like this. We actually get really precise measurements of how much DNA is within each nucleus of each cell. It corresponds to the size here. And we see these beautiful peaks that represent the natural cell state, and then it doubles its DNA. And then this cell splits in two, and it goes back to the original size. So these are twofold different from each other. And as you can see for this, the goal is to find a compound that causes the cells to then quadruple or so on. So 4 times, 8 times, 16 times the amount of DNA. And that's what we did in this particular experiment. We're proud of this one because it's very rare in academia that we get to push something to a clinical trial, but we were very lucky to find a compound that already was approved for use in humans, and it looked effective for myelofibrosis. So John Crispino, who is leading this project, was able to start a clinical trial and then successfully treat patients with the compound that we found in the screen. And then lastly, imaging, another reason we love microscopy is that it's highly multiplex. You can measure lots of things at one time using image-based data. And we'll talk a little bit later about the cell painting assay, which allows us to use different fluorescent channels, label different things in each channel so that you get a multicolor picture revealing lots of things about the cells. Okay, so drug discovery needs fixing. We think microscopy is a great way to accomplish this together with computer science. So now I want to turn to a little bit more closely what our lab is working on, and that is a revolution in how we use images. So what is, in the old days, when I was a graduate student, this is now 25 years ago or so, we had microscopes and they were starting to be automated. They were starting to have robotics attached to them, but the computational software to control them was pretty primitive, and the image analysis was also quite primitive at the time. Nevertheless, given that I grew up on a farm, it was still better than farm work to operate one of these microscopes, even if you had to do it manually. So that was during my PhD, but as I got towards my postdoc, I really felt that this is something that needs solving. So here's a problem that needs solving. We need software that can analyze images so that it's not a manual process for biologists to look at images by eye. So for some of you, this may be difficult to believe that back in the old days, 25 years ago, it was very common for biologists to capture thousands and thousands of images manually by looking through the microscope and clicking a button to capture the image, and then quantifying the phenotype also manually by just judging, are the cells kind of big, kind of small? Are they green? Are they bright green or medium green or light green? Those kinds of phenotypes, the biologists would literally just, write down in a notebook. So that's clearly something that needed solving, and that's why Cell Profiler was born. This is the software that I wrote during my postdoc. Started writing in MATLAB originally, now it's in Python. And together with Thoas Ray Jones, he was a computer scientist student at MIT at the time, we put together this software to really solve this problem for biologists. It's open source, of course, and really focused on high throughput image analysis so that people could make a modular pipeline that would solve their image analysis problem, and then they could run it on large scale experiments. So this was right at the transition time where people were moving from doing an experiment that involved capturing 10 images to an experiment where they tested hundreds to thousands of different samples and really needed to quantify them. So I say thanks to the snowstorms because I was a biologist at the time, of course, but I spent that first winter, as shown here, just a lot of snow. This is a list of the, sorry, the arrow's in the wrong place. This is the winter that I wrote most of Cell Profiler because we had so much snow in Boston that I just pretty much stayed in my apartment and wrote the software. The software has been very successful. It's cited in a few thousand papers every year. Biologists really feel warm and fuzzy about using the software. It makes it easy for them to analyze their images. Most pharma companies use it for drug discovery. And I just put this is maybe the most important takeaway, especially for the biologists in the audience who want to do an image analysis problem. There's a beautiful online question and answer forum, not just for Cell Profiler, but we created it for the whole community to ask any question about open source image analysis software. So if you're stuck on an image analysis problem, post something there and you'll get really careful and helpful responses from the whole community. And I do want to say as well, this open source software project has typically only had a single software engineer working on it over time. Originally, it was myself with a bit of help from Ray Jones, then it was mostly Ray. And you can see all the contributors over time. Of course, many more contributors than what is shown here, but largely it's been one person over the history of the software. And it just shows the impact that you can have without necessarily an entire software team. Okay, so the informatics advanced during this time. The other thing that advanced over 20 years ago or so is the automation to do these kinds of experiments. So in the old days, you might literally have a petri dish with cells growing in it, and you might add a compound to it. Instead, we now usually use these 384 well plates. So there's all these rows and columns of little plates. On the bottom of each one, it's see-through so that the microscope can look at the cells that are growing on the bottom of each of these individual wells. And now we can test 384 chemicals in a single reasonably sized plate that we put on the microscope. The microscope is automated, adding the compounds is automated, and so this has really sped up a lot of things in the field. And so that brings it all together. Now we have the robotics, we have the informatics, and we can do sample preparation for hundreds of plates, with each of them having hundreds of samples. So we get into the millions in the pharma companies who do these kinds of experiments. We have the automated microscopy. This can continue without any manual intervention. We get thousands of images from every plate because we take more than one image per well. The images can have many channels, many different colors present because we can label different things. And then we carry out the image analysis and the segmentation. Cell Profiler usually, but we often are using Deep Profiler, a deep learning-based software for segmenting and extracting features. So once we've identified the different compartments of the cell or the different cells that we're interested in, it's trivial to measure thousands of features for every cell. And the way that we get to thousands of features is because we measure a bunch of categories. Each of these has a few dozen metrics within the category. And then we measure each of those categories against each of the channels in the image. So it's these, let's see, 7 categories times 5 channels times 2 or 3 different compartments or regions of the cell and so on. So everything combinatorially adds up so that we have thousands of features per cell. Okay, so now we get to a revolution in how we are using images. I've taken you through a bit of my career path and history to understand how valuable this automation of the image analysis and the robotics has been to get us to the point where we have lots of images. What do we do with these images in order to accelerate drug discovery? So we'll talk first about a field called high content screening. You can ignore these terms, these individual terms. The concept is in the old days, when I started in my career, our goal in doing image analysis was to measure known phenotypes that the biologists cared about, because they know if I measure this thing, I can potentially cure this disease. And we're transitioning to a world where we can profile samples. We measure all their features in order to characterize them. So let's start here with measuring known phenotypes, and I'll show you some examples of how this accelerates drug discovery. So here, lots of different kinds of biology are shown. I'm not going to go into detail of any of them. It's really just about, let's look at, let's just see what these look like. So we can measure worms. There's an example here at the first and last examples here involve entire worms growing in wells being treated with different chemicals to see which ones might have the certain outcome that the biologists want. So in each case, the biologists worked with us and they said, we want to find genes or compound perturbations that cause these DNA damage speckles to go away, or that cause this green protein at the tail of the worm to extend throughout the worm, or that cause these cells that normally look like this to look like this. So in each case, the biologist understands the disease area, they understand the stains that they're working with, and their goal is to measure a particular thing. And there's lots of different examples just to give you a sense of the kinds of things. We're measuring intensities, the shapes of things, the colors, the relationships between colors, and so on. And this covers all kinds of different disease areas where we can help people find drugs or genes that, when perturbed, they impact the phenotype, the outcome that the biologist cares about. And these kinds of images can also be multidimensional. They can be 2D. 2D is what we typically work with, but they can also be 3D or time-lapse. And so that's some examples that are shown here as well. This kind of intentional measuring a particular outcome of cells has been really successful. And so the entire pharma industry uses image-based readouts now. For at least the past decade, it's been quite common. And so they're discovering all kinds of drugs every day. The project that we were involved with, I already explained to you and found a useful cancer drug. Here's a project we weren't involved with, but where they used our software Cell Profiler in a clinical trial. They took tumors out of patients, grew them in a dish, separated them into one of those plates so they could test 384 compounds, and tested different known cancer drugs on the patient's individual tumor. So instead of giving the patient your best guess compound, which is how doctors usually have to do this, they instead tested a few hundred compounds to say which compound actually kills this patient's tumor, and then gave the patient that particular compound. And this was a very successful personalized medicine trial, and I hope will become more of a standard for people who have cancer. Most of what I've described up until now used classical image processing, so mathematical algorithms that that process images in particular defined ways. But as everybody knows by now, deep learning has really taken over the world, taken over many different fields, and that is also true for biology. It's starting to become more useful and in practical biology experiments. One of the first experiments we did was to organize the community and gather up lots and lots of images. We outlined 30,000 individual nuclei that are shown here in order to allow people to try to train algorithms, not just to improve the accuracy on a single data set, but our goal here for the first time was to achieve generalizability. Within the biology domain, it was constrained to let's just make a model that can identify any nucleus in any image with any sort of weird stain that the biologist might find. So the nuclei, you know, here are the purplish things. Here they're also kind of purple, but obviously a very different setting in the, with pink, this pink background. And here there are these black dots in amongst a bunch of other clutter and so on. So they can look quite different from image to image, but if you put them in front of a biologist, or really anyone, if you train them for 5 minutes to understand what a nucleus looks like, it's not a terribly hard problem for humans to do, but at the time, completely impossible for algorithms to be able to be flexible and generalizable like this. So it's a really cool project. Well, the outlining was not very fun, but we did it, and we got thousands of teams competing in this competition. And if you compare it to a classical tuned algorithm that takes a lot of hands-on time to set up for each individual batch of images, these methods can do not only better in terms of accuracy, but require exactly 0 tuning for each individual data set. And of course, advancements have been made since this time, so it's a really exciting time for deep learning in biology. My lab doesn't work on segmentation problems anymore. Instead, our alum, Beth Simony, has taken on this area of research and is working on deep learning powered tools for segmentation, but also classification of samples through an NIH funded center, as shown here. So that work definitely continues, and it's a really exciting area of biology image analysis that is ongoing. But as I said, my lab has moved away from segmentation and now is working on image-based profiling. So instead of measuring exactly what the biologist tells us to measure because they care about a particular outcome, we measure everything we can from each biology sample, and then we do something with that information. So it says, please turn on your pointer on your slides. Oh, you can't see where I'm gesturing, it sounds like. I don't know that I can do anything about that, unfortunately. I don't know how. Okay. Okay, thanks. Okay, so the whole rest of the talk is going to be about image-based profiling. So if you've gotten a little sleepy, now is the time to wake up, because it's important to understand this slide to really kind of get the concepts of the rest of the talk. So image-based profiling is where we use images to create signatures or a pattern that comes from different perturbations, genes or compounds or diseases. So let's say we're studying drugs. We have drug number 1, drug number 2, drug number 3. We add them to cells. We stain the cells in an assay that I'll describe in a moment. They look different from each other by eye, but of course we don't want to be looking at things by eye because there's so many samples. So we carry out image analysis, and we end up with these beautiful data matrices of thousands of cells measured for thousands of features. And this is just a tiny fraction of a real experiment. You know, I'm only showing a few hundred of each for 3 samples, but we might literally have 100,000 perturbations, many replicate samples for each one, and then thousands of individual cells for each individual one. So huge, huge data matrices coming out of these experiments. But the fundamental concept is we just want to see, is there a difference between the profiles that come from drug 1, drug 2, drug 3? And as you can see, there's heterogeneity, of course, but we often can see patterns in this kind of data. And then we take that data and we measure similarities and connections among the different samples. And I'll show you what the actual concrete outcome of those kinds of experiments can be in all the rest of the examples in the talk. If you're familiar with transcription profiling or proteomic profiling, image-based profiling is exactly the same. We extract the features, and then everything downstream is much like these other profiling fields. Okay, you might ask, well, in contrast to the first half of the talk where we were talking about particular biology outcomes that the biologists care about, here, if we're measuring everything, what does that mean? What do we stain the cells for if we just want to measure the general properties of the cell? And we chose to label the major organelles within the cell, the major subcellular parts of the cell, with these different dyes. And these dyes are really cheap, and that makes this a very inexpensive assay, much cheaper than transcriptomics or proteomics, and giving us single cell data where we can measure thousands of samples really inexpensively. So this is what the assay looks like. Again, we're measuring size, shape, intensity, texture, and so on across all the different colors, the channels of the image, and all the different compartments of the cell. Okay, so now here's the kind of schematic of the drug discovery process. And we're going to now look at individual examples along the way. So if you miss anyone or it doesn't make sense, they don't build on each other. They're just a bunch of individual stories about how this process can, how the microscopy and computer science can accelerate each of these different kinds of applications within drug discovery. So discovering a drug is a multi-step process. We have to develop some kind of assay. We have to test lots of drugs against that assay in a big screen. Then we have to optimize the compounds, put them into preclinical studies and clinical trials before we know that we get an effective drug. So let's look at this first one. Can we identify the signatures of a disease? And if we can screen drugs to reverse the signature. And here we have an example from the University of Utah. They eventually formed a company, and I serve on their scientific advisory board. What they did is they were studying a particular disorder called CCM, and it causes strokes in humans. And they found if they perturb the gene that is known to cause CCM, the cells look different. So healthy cells look like this. They knock down this gene that causes the disorder, and the cells clearly look very weird. You don't need fancy image analysis to tell the difference between these two. But by setting up the experiment in this way, what they can do is now take cells that are in this diseased, unhealthy state and test drugs one by one. Are there any drugs that cause these unhealthy cells to look more healthy again? And they carried out this experiment and found that if they used computational analysis to analyze the images, the drugs that they found were much more effective in some downstream experiments. Eventually, they tested mice that have this disorder to see whether the drugs would help them. And they now have a clinical trial launched for this disorder as well as several others, because they've taken this same approach for lots of different diseases, thousands of different diseases, and now can screen them all in parallel in very high throughput. So this is one of the first examples of a company that has parallelized drug discovery and kind of made it systematic, where instead of having a team of hundreds of researchers that study the disease for decades, instead they set up these high throughput experiments that they don't cure all of the, they will not yield cures for all of the diseases, but for whatever fraction is low-hanging fruit, it's a really nice systematic approach. Back in my own academic lab, we have been working with the University of Toronto on a project where they, I'll explain how this project works. Again, the goal is to find the difference between diseased and healthy cells, but instead of knocking down the gene, here we labeled the gene, the gene's product, which is the protein, and it looks a certain way. So here's this gene called LZTR1. I don't know what it does, but this just shows us how it's located. So I don't need to know what it does. All I need to know is that when I make a particular mutation, this is the name of the mutation within this gene that is associated with patients who have Noonan syndrome. When patients have Noonan syndrome, they have this mutation and it causes the protein to just have a different location within the cell. And you don't need to know anything about the disease or the gene. All you need to know is any image analysis software should be able to tell the difference between the left and the right. Just like a pathologist who's trained could tell the difference between a healthy boy and a boy who has this syndrome. I can't tell the difference, but a trained pathologist can. Here, we're using cells, and it turns out the difference is very clear, even to a non-expert. And what's lovely about that is, well, we can't do experiments on the humans, but we can do experiments on these cells growing in a dish. So now let's take cells that have Noonan syndrome and test drugs to reverse this phenotype. Now, this doesn't always work. 90% of the time, when we make a mutation that causes a human disease, the cells look exactly the same. So we can't do anything, it's useless. But for that small fraction, that less than 10% of the time, we do see this kind of a change. And we are able, so far we've been able to find a couple hundred disease phenotypes this way. And each one of those represents an opportunity to test drugs and potentially find a treatment for these disorders. Now, 200 is what we found so far. Here's some examples of some of these others where we have the healthy state and the disease state for lots of different diseases. And you can see the gene names and the mutation names are all shown up here. But many of these are very visible by eye. Some of them are very subtle, and we need fancier image analysis to tell the difference. So far, we've discovered 200 phenotypes from screening 3,000 variants. But we just got funded through the NIH to carry out a much larger experiment where we're going to have tons and tons of these coming through. So we have tons of data heading our way over the next 3 years, where we will be discovering many more. And the next step will be doing drug screens on most of these. Okay, so that's one way to identify a disease phenotype, is knocking down the gene or tagging the gene. A third way is to just take cells from patients that have the disorder. So for bipolar disorder and other diseases of psychosis, it's a little harder to say this gene causes the disease. There's many genes that contribute, but none of them are very causal. And so instead we take samples of skin cells from patients, we grow them in a dish, and we discovered, this is an experiment going all the way back to the time when Mohammed was in the lab. We've been working on this quite a long time, gathering the cells. We find that healthy patients have these red mitochondria really stretched out throughout the whole cell, all the way towards the edge. And by contrast, patients that have bipolar disorder, they are much more likely to have the mitochondria squished towards the middle of the cell, not spread out towards the edge. And because we've identified this in the patient fibroblasts, we can now take the patients that are in the unhealthy state and test drugs to try to find some that reverse morphology. So we are now collaborating with the NIH to do an experiment on these cells and hopefully find new drugs for this set of disorders, which is very difficult and painful to deal with otherwise. Here's just some data on those different mental conditions. Control patients are shown here with respect to this metric of mitochondrial dispersion, how far away the mitochondria are from the cell. So these are healthy patients here in light blue. And then these reddish, pinkish, purplish ones are all the different diseases of psychosis. So you can see it's not completely separable. This is not a good diagnostic. However, we do see that these patients on the whole have mitochondria that have a different location than typical. And again, we can test drugs to reverse that morphology. All right, next example is, can we identify gene and allele functions by grouping similar genes and alleles together? And here, again, going back to the days when Mohammed was in our lab, we overexpress each gene. It means just like we sort of add extra amounts of the gene to each cell. We do the cell painting assay, the image analysis that we discussed. And then we cluster those morphological profiles so that images that look alike are nearby each other, and therefore the genes that they represent are nearby each other. And I hope you can see to some degree that we see all these different biology pathways. So it took decades for biologists to identify each of these different clusters and pathways. Many of these are already known, but you can at the very least see that we have replicates. Replicates land near each other. Similar genes, RAF1 and BRAF, are very similar, as is KRAS. They all have kind of related names because they're similar. This is the hippo pathway down here, two proteins that are known to physically interact. So we were able to identify a lot of known biology and even uncover some new biology just by asking, when you mess with this gene, do the images of the cells look alike? There's a lot of information present there. So this is pretty exciting and caused us to scale up in a lot of ways. One way was to do a knockout of the entire genome. So each gene, 20,000 genes, one by one, knock it down, and ask whether the morphology of the cells has changed. And we did this in a pooled format in collaboration with all the folks shown here. And that allows us to kind of make a map of the morphology of the cell, where genes that are known to have similar functions tend to cluster nearby each other in this map of morphology space. So really cool kind of experiment that allows us to determine the function of some genes that are unknown. It might surprise you, for those who aren't in biology, that we don't know what all the genes do yet. And so this kind of an experiment gets us closer to identifying that. Another huge source of data we have is just an exciting time for the field, because up until now we've had just a few hundred samples here, a few hundred samples there. Just in the past year or so, we've ended up with tens of thousands of samples through the experiment I just showed, as well as this huge JUMP consortium where we managed to get 10 pharma companies together with a couple nonprofits to create the world's largest cell painting data set, which was just publicly released and is now available for anyone to mine and take care of. We're still doing batch correction and normalization and plate layout correction and so on, but really excited to make this data available for the community to work in this space. All right, I'll just show a few more examples. Can we identify chemical regulators of a gene profile? So by profiling genes, we can figure out new gene functions, but we can also identify chemical regulators that might be good drugs for diseases that are caused by a particular gene. And this project involved, we did this with a fairly small set of data that was available at the time. We just had 100 or so genes. We tested 69 of them, where you perturb the gene, you get some interesting signature. So some of the features go down, some of the features go up, and this is the profile of that particular gene. And you say, I've got all this big set of compounds. Do any of the compounds match exactly this gene readout? And this third one down is a pretty good match for it. And there's other samples that oppose the signature. They have exactly the opposite signature. And so we ask, do those compounds, do they impact the function of the gene? Are they known to be associated with the gene? So first we tested this with positive controls, like ground truth. We knew that these genes and compounds were related. And we found 32% of the time, if we used a gene as a query, we could get back a compound that is known to impact that gene. So that was pretty exciting. We're really thrilled to see this. And but you don't really trust anything until you can do it prospectively, because there's all kinds of ways to overfit or fool yourself. So we did a prospective test where we took 7 genes that had compounds to match them. And we asked our biology collaborators to go and physically test those compounds in whatever assay they wanted to test. And depending how you count, 3 to 5 of those genes were successful. So definitely matching or exceeding the retrospective rate. And in particular, we found regulators of YAP1 that's in the Hippo pathway, which is really exciting because it found pathway regulators that kill sarcoma cells, which is a very untreatable kind of cancer right now. Usually there's no drugs that are specific for sarcoma. So we're really excited to pursue those and develop them and see if they might be able to go to clinical trials. Can we, I think 2 more stories. Can we assess the mechanism of potential drugs based on their profiles? And here we have a comparison between image-based profiles and mRNA-based profiles. For a lot of folks, it's really hard to believe that images can, just the morphology of cells can really tell you that much about what cells are doing, as compared to mRNA, which feels comprehensive because you're usually measuring thousands of mRNA levels, and it just feels more quantitative and so on. So I'll show you this next experiment where we tested them side by side. This is just one little anecdote. You shouldn't conclude anything from this. It's just to demonstrate that when you have image-based data, this is cell painting data here, and you have a low dose of the drug, you get kind of messy profiles in all the replicates. But then as you increase the dose, you end up getting a nice, strong phenotype that when this drug is added, the cells look a certain way by imaging. Same thing can happen by mRNA profiling, and this L1000 is an assay for mRNA profiling, and this one just happens to be a little weaker by mRNA profiling, but you still see a signature here. So that's what we're trying to do. We're trying to say, if I have a drug and I don't know how it works, can I do this kind of an experiment and get this kind of a profile and match it against other drugs that have known phenotypes? So that's what was done in this experiment, where we compared the mRNA profiling with the image-based profiling, cell painting. And we do this along a dose response curve. So higher doses give you more good matching of the compound mechanisms. But I'll focus really just on this last bar where we allow every dose to match every other dose. So what's happening in this last bar here is the pink part shows us out of all the assays we tested, 34 of them were really nicely effective using both mRNA profiling and cell painting. Either one of them does a really nice job of taking compounds that are known to work in a certain way and grouping them together. But you can also see that there's some assays that only mRNA profiling is revealing the mechanism of those drugs. And there's other assays where only the imaging is revealing the mechanism of the drug. And you can see that a little more clearly. This is an unsupervised analysis. This is a supervised analysis where cell painting does a bit better. But the point is, some different kinds of mechanisms of compounds are very nicely discovered by both assays, and others are more effective in one versus the other. All right, the last story. Can we predict the outcome of a complex assay by correlating its readout to cell painting profiles? Here, I'll just skip ahead to here. Here, it's helpful to know, and this is where we really get to AI and drug discovery. So to me, it blows my mind that this kind of an experiment is possible. If you give a deep learning network, we're not talking about images here, we're just talking about chemical structures. If you give a deep learning network some chemical structures and you train it, you can then give it a new chemical structure and say, hey, deep learning network, is this chemical likely to be giving me a good outcome in assay number 1 or assay number 2 or 3 or 4? And if with enough training data, you can get decent results here. So I guess by decent results, I should clarify, it's on the order of maybe 5 or 6% of the assays. You can actually create a deep learning model where the chemical structure alone tells you whether that chemical is going to be a good hit in the assay, whether it's a prospective drug for that particular disease that the biologist is testing. So we took hundreds of these assay readouts, we trained models with the chemical structure alone, but that really feels unfair because the chemical structure hardly gives you any information about what the drug is actually doing. We should give a little information about how that drug impacts cells. So let's also give the network images as well as mRNA profiles that show the impact of the drug on cells, because that really should be helpful. And it is helpful. Here's how the results look. We get out of there's 270 assays total. So again, fractionally, this is a pretty small fraction of the time that this works. But each assay that we can predict using a deep learning network is an assay that we don't have to perform in real life. Each assay can cost $5 million, $10 million in a pharma company environment. So even if we can predict 5% of them and do them virtually, computationally, rather than in real life, that's a huge win. So for chemical structure alone, 11 out of 270 were predicted using just chemical structure. Similarly for gene expression, morphology was a bit higher. And then here's all the numbers total. If you just have one of the modalities, morphology gives you the best predictions. If you are allowed to do data fusion, taking all 3 modalities together, you actually don't do any better, which to me says there has to be more information present in these other modalities. To me, this says data fusion methods need improvement. And then if you also do kind of an oracle retrospective analysis, you can, in theory, get even higher. So 28 is starting to get to be 10% of the assays, which is really, again, has a huge monetary impact on pharma companies. So our lab is not just working on these different applications. We work on a fair number of the methodologies that underlie them, things like batch correction. And here's an example of that. I won't go into great detail, but as you can imagine, there are lots of details under the surface of exactly how do we do segmentation, how do we normalize, how do we do batch correction, and so on. That's really important to get the good outcomes that we're looking for on the application areas. So believe it or not, I only showed you a fraction of the kinds of things images are being used for in drug discovery today. This slide gives you many more examples, as well as the place to download these slides. And with that, I really thank you for your attention, and I'm really happy to take questions about any part of the talk.
So thank you very much for the interesting talk that you had. And let's start the question part. There are some questions that audience asked, and I will ask them one by one. As first question, is it possible to count the rate of MN in micronucleus assay with cell profiler or not?
Is it possible to, I didn't quite hear.
Count the rates of MN in micronucleus assay with cell profiler or not?
Count the rates of, I didn't quite hear the.
MN in micronucleus.
MN micronuclei, is that what you're talking about?
Yeah.
Oh, yeah. So for those who don't know, every cell has a nucleus. You can see a picture of that here. In the middle of the cell, you have the nucleus. And a micronucleus is where a little chunk of the DNA kind of blebs off. The cell doesn't divide properly and ends up with a micronucleus, a teeny little chunk of DNA. That's bad in general. You don't want drugs that do that to your cells, because then you end up with all kinds of nasty mutations and probably cancer. And so micronucleus assay is trying to measure those little blebs. And yes, those are pretty easy to detect. Sometimes just with regular segmentation, you can identify small blobs and count them. But certainly with machine learning, to identify those little blebs, it's a very solvable problem with image analysis these days.
Yes. So for the second question, in your opinion, how do you see the importance of radiation biology for drug development, especially radio sensitizers and radioprotectors for clinical use in radiotherapy or brachytherapy, with the help of DL for tissue studies focusing on diagnostic images like MRI for drug development?
Okay, sorry, I'm sorry to make you repeat it, but I caught like half of the words. So just one more time, please.
All voice is not proper, but I will repeat that in louder voice. So in your opinion, how do you see the importance of radiation biology for drug development, especially radio sensitizers and radioprotectors for clinical use in radiotherapy or brachytherapy?
Okay, yeah, let me close that curtain so it's less visible. So I don't know much about radiation oncology and cancer plus radiation. I mostly focus on the drug side, but it does seem to be a growing interest to say, instead of just, okay, there's 3 ways to treat cancer. You can cut the tumor out and hope it doesn't grow back. You can do that, and also radiation to kind of mess up the cells at that one site so badly that they don't want to divide anymore. And both of those can be very uncomfortable and possibly toxic. And then you can give drugs, and those drugs can be conventional drugs, or they can be immunotherapy, which some of you may have heard of for cancer, and so on. And so one way to make radiation less toxic to the patient is to give the tumor some kind of sensitizer, some kind of material that localizes right at the tumor and makes just the tumor more sensitive to radiation. So the rest of your body, you can use a higher dose of radiation or a lower dose of radiation that makes the rest of the body not so toxic and happy. And then it's like really targeting it. And so it's a cool approach, but I don't have any I don't have any expertise to say whether it seems promising is all I can say.
Okay. And for the third question, in the test compound, if one drug killed the tumor, is it still effective on the bigger scale, which is the tumor itself, considering out test case is a really minor case?
Is a really what case?
A really minor case.
I'm sorry, can you repeat the whole question then again?
I think he thinks that if the test compound, if the test for example a particular compound, it is still effective on a bigger scale, which the tumor itself, considering out test case, is a really minor case or not?
Yeah, I'm sorry. I guess I don't really understand the question very much. It's outside my expertise for sure. But I guess yeah, I don't know if I have anything useful to say about it. I'm sorry.
I think, but okay, let's skip this question too. And I myself have another question. Is there any practical test by using drugs for reversing the phenotype based on this method? I mean, in practical, in a practical experiment in labs or something. By drug for real cases like not humans, but other animals for tests?
Yeah, so sorry, I'm gonna, hopefully I'm answering the questions. It's a little hard to understand, but typically the next step after drugs look promising, so we usually test drugs in human cells growing in a dish, and if they look promising there, then right now most regulatory agencies require that you test the compounds in at least 2 animal models, 2 different kinds of animals. So to me that's quite surprising. I didn't realize this. So it's not just mice or rats, but mouse or rat or dog or monkey, and those have differing similarities to humans. It's not perfect. So the reality is we often rule out drugs that might work well in humans, but they fail in some other animal, and so they never go to clinical trials. So there's a big movement right now to stop using animals in testing, not just because of the ethics of it's not just because people care about animals, but rather because the animals are not very accurate to predict effects in humans. So that is a step that happens right now, animal testing, but it's increasingly considered to be not ideal.
Yeah, and last question is, it's a question for starting career in this topic. He said that what scientific and educational path do you suggest for a master's student in artificial intelligence who is interested in working in the field? He means drug discovery, actually. In fact, what skills are needed to start research in this field for this group of people?
Yeah, I love this question. So in general, I would say it's going to be increasingly valuable to have some domain expertise. You don't have to pick biology, but if you are working in computer science, it's going to be extremely valuable to have some domain expertise in some other area. I think biology is a great area to choose because it's going to be really growing over the next few decades, this intersection of AI and biology. So I think it's a very valuable one. So then the question is specifically asking, what do you do to kind of get more involved? And I think there is some value in taking actual coursework. It doesn't have to be a formal course. It could be an online video series or something like this. I think there's some value to getting a big picture overview of biology to get a general sense of the field. But I think fundamentally, you really have to just start working on biology problems. And I will warn you that if you've not worked in biology and you're in computer science, it's really messy and frustrating. I've made these examples I showed today sound very straightforward, like, oh, you just overexpress the gene and then this happens. But when you really get into a problem, you understand how frustrating and messy biology is. We can't always predict everything. We try to turn genes on, but they don't actually always turn on, and we try to knock them down and they don't always behave the way they're supposed to. So I think working hands-on projects in a given domain is really the best way to start to understand more about the constraints of the field.
Mm-hmm. And so that's the end of the questions. Thank you very much again for the interesting talk that you had, and thank you for joining us and accepting the invitation.
It's been my pleasure. Thanks, everyone.
Goodbye for now.